So in this short presentation, I just want to talk to you about how to prepare for your upcoming centre assessed paper in biology, GCSE biology. You're going to be sitting a paper 1. The real exam is 1 hour 45 minutes, so that will be reflected in this exam as well. Okay, it's a little bit longer than the students that do the double award paper, but you're sitting triple award, so yours is 1 hour 45 minutes. Before you go into the exam, you need to make sure you know where you're sitting. You need to make sure that you are properly equipped. So you're going to need a black or a blue pen, plus a spare in case it runs out, a pencil, a ruler, a rubber, and a calculator. Definitely make sure you have a pencil and a ruler for graph drawing and for drawing labels. And a calculator is an essential piece of equipment as well. You will definitely need that in your biology exam for calculations. So what do you need to revise? I've listed absolutely everything from the revision guide on this particular slide here. I'm not going to sit here and read through it all, but it is cell structure and cell division, okay, as well as movement in and out of cells. Organization topic, in which you've got the lungs, the heart, you've got digestion, and you've got plants in there as well. Please don't forget about plants. Infection and response. Okay, so communicable diseases, bacterial, fungal, and viral infections, as well as how our body defends itself against all of those. And then we've got drugs, so we've got antibiotics, we've got vaccination, the development of drugs, and something that is specific to you triple award students, monoclonal antibodies, and plant diseases and defense. Lastly, you've got bioenergetics, okay, so photosynthesis and respiration. The inverse square law is something that comes up for you triple award scientists, so please make sure you look at that, okay, and how we can control the rate of photosynthesis, so making plants grow as quick as possible and as big as possible, okay. And for respiration, don't forget you've got aerobic and anaerobic, and you need to know the effects of exercise on the body as well. So first thing you need to do when you start your revision is work out what you already remember. Okay, now what you've got to remember is that some of this you did in year 9, so you haven't touched this for quite some time. Don't be surprised if you have forgotten quite a lot. Okay, if you've got your revision guide, that's a good place to start, but there's also lots of GCSE revision videos on the internet. So a good technique to use is self-quizzing. Okay, so open your revision guide to the page, the topic that you want to revise that day, and fold a piece of A4 paper in half lengthways from the top to the bottom. What you need to do is, as you revise that page in your revision guide, is you need to turn the information on those pages into a series of questions. So on one side of the piece of paper, write the questions, and on the other side of the piece of paper, write the answers to those questions. Then what you need to do is fold the paper in half, so you hide the answers, test yourself, and if you get any of the questions wrong once you go through them, highlight those and revise those sections again. What you're doing there is you're identifying areas of strength and identifying areas of weakness and what you need to revise again. You then need to try some practice questions. You do get plenty of these from homework anyway from your teachers, but if you want to try more on your own, that's brilliant. If you Google search AQA past papers, a series of drop-down menus will appear. Okay, so you sit AQA, make sure you get the right question papers. A series of drop-down menus will appear, and you can select GCSE, not A-level. Make sure you select GCSE, and you select biology from those. And they're PDF documents that you can download. You can try the full past paper. You can time yourself, or you can select individual questions from there. But the most important thing is, once you've tried these questions, is to work with the mark schemes. Look for keywords that are essential in the answer. If it's in bold or if it's underlined, it must be in your answer. Look for things that are not acceptable. Sometimes, even though you would give a verbal answer to a teacher in a lesson and we would know what you mean, writing that in an exam might not get you a mark. So look at what's acceptable and what's not acceptable. Work with the mark schemes to spot patterns, okay? And the key thing with biology is keywords, making sure you hit the nail on the head, get that mark by including that particular keyword in your answer. And you're going to get all this information through looking closely at those mark schemes. Right then, a few specific questions to look at, and a few little exam techniques. Okay, the first thing, it may seem like a really simple instruction, is just make sure you read the instructions carefully in the question. If you're asked just to tick one box, don't tick more than one because you won't get any marks. They're not just going to award you the mark for the correct one and then ignore the one that you filled in extra. You're going to lose the mark. Okay. If you are asked to draw one line from a box to another box to a corresponding box, make sure there is just one line. And if you draw a line and it's incorrect, scribble it out carefully or write a little label on saying it's this one for the correct answer. Make sure your answer is obvious. Magnification, so microscopes, microscopy comes up in cells in the very first unit of paper 1. A couple of things, make sure you know your formula triangle. So IAM, it's easy enough to remember, image, actual, and magnification. Make sure you know how to use the formula triangle with covering the one up that you want to find. Look carefully at the measurements on any diagrams in the picture. So if the length is given in micrometers, you're going to need to give your final answer in micrometers. If it says give your answer to a certain number of significant figures, make sure you do that as well because it will carry a mark. Remember, units are always important on the end. Okay, magnification doesn't have any units, but if you're talking about image or actual size, you're going to need to give it 9 times out of 10 in micrometers. It's a U with a funny stick on the front and then a little M. And remember, 1 micrometer is 1,000th of a millimeter. So to convert between millimeters, which you will do when you place your ruler on the diagram to measure a particular line or a scale bar, and then you need to multiply that by 1,000 to convert it into micrometers. When it comes to making measurements on the paper, that's why you definitely need a ruler in a biology exam. Okay, measure in millimeters and go right the way to the ends of the arrow. Most mark schemes will give a little bit of leeway either way, just to account for students, going for the beginning or the end of the arrow, but go right the way to the end of the arrow. Describe and explain. So some students really struggle when it comes to graphs answering the question correctly. Okay, it may seem really simple, the difference between describe and explain, but even at A-level, students still make these mistakes. So in this particular graph here, you've been asked to describe the changes in the number of live cells shown in the graph above in the first 20 hours. So describe is say what you see, what happens. Look at the number of marks available, and if there is more than one change in the graph, describe each change in the graph. Look at the number of marks available, and have you made that many relevant points about the graph? Describe the gradient of the line. Is it steep? Is it not steep? Okay, does the rate increase? Does it decrease? Look for every change in the graph and point out every piece of information about that graph. Describe is say what you see. What can you tell about that graph just by looking at it? Okay, and one key point there is where it says use data from the graph. Make sure you refer to some numbers from the graph. If the rate starts to increase, at what time does the rate start to increase? Does it increase sharply? Does the rate slow down at any particular time? Pick data out from the graph. Explaining is different to that. Explaining is what is the science behind the shape of the graph, or in this example here, the information in the table. So in this particular question, you were asked to look at the digestion of some fats, and you were looking at the change in the pH of the solution in a conical flask. Now, the pH is going to be altered because of the fatty acids that would be present in the solution. Now, it says, give one reason for each of the following. That is an explanation question. And it says, use information from table 2 and the graph above. Obviously, you would be able to see a graph in the question. But you need to then explain why is the pH decreasing over time. So it's the science behind it. It's not this is the pH decreases. It's asking you why. So when you see explain, it's explain why the graph is that shape, or explain why the results show that particular pattern. What is it that's about that particular investigation that is causing the change in the data? Explain why. You may be asked to draw a graph in a biology paper. There usually is one graph. These usually are 3 or 4 marks. This particular one here is quite nice because it tells you what it wants you to do. It tells you it wants you to label the axes. It may not give you that particular instruction, and you may need to realize you need to do that. But make sure that your independent variable goes on the x-axis and your dependent variable goes on the y-axis. Make sure you include the correct labels and the units. Don't forget the units. Plot each data point and then draw a line of best fit. Make sure your scales on your x and your y-axis are good. Don't want your graph all bunched up in one particular corner, or you don't want it. You want each space on your y-axis and your x-axis to be worth the same value. Check your scales before you draw. Don't use massive crosses, use little crosses or little points that are obvious to see. And then please don't forget, you need a line of best fit. Okay, we do not join points together. We draw a line of best fit, which is again why you need a ruler in a science exam. Okay, if you need any help, get in touch with your teacher, either through Teams or you can speak to them in lesson, but you need to make sure that you are properly preparing for these exams that begin the week beginning the 16th of November.